Hello, this is Dr. Paul Cottrell, and I am recording this on 3/13/2025. I had a subscriber on my Patreon channel asking for me to go over self-replicating RNA. So, their request was a couple months ago, or maybe more, and now I have some time to do that. So, this is for you, for the one that asked for this. Please help support my work by making sure you subscribe to my different channels. I have 7 channels on YouTube. I have Brighteon, Bitchute, and Rumble. All the links are in the description of this video. Please click those links and subscribe. It's really important. I live stream on YouTube. I also live stream on Rumble. You get to see replays, replayed premieres on the different channels, so you can interact. I also do live um, X spaces call-ins. So there's quite of interactiveness that I've built into this show. So please help support my work by making sure you subscribe. It's through the donations and the ones that are paid members to Patreon that help support my work, and also the ones that purchase products on my store. So please take a look at the products that I offer and help support my work. Please make sure you subscribe to all the channels and ask your social media to follow me. There's a lot of censorship that's going on YouTube, and every time I post, there's a bleeding of my subscribers. So please help support my work. It's really important that I reach a larger audience so I can inform people on how to navigate through the different problems that they may be seeing, either with their personal health or with what's going on in the financial markets or what's going on with politics or international affairs. So it's really important that I reach a larger audience so I can help as many people as possible. So, with that said, you can donate on my website, the-studio-reykjavik.com. The link is in the description of this video. You can donate through Stripe, PayPal, or buy me a coffee, or you can be a paid member to my Patreon channel. It's a great way to help support all these videos that I do for free. I don't put anything behind a paywall. And also, you can watch the live broadcasts or the audio podcasts. So you can listen to the radio section, or you can listen to the live streams that are backed up that you can replay on my website. So it's a great way to help support my work, and then you can get the different products that I offer. So please help support by going to my store, the-studio-reykjavik.com, and share the links. Ask your social media to follow me. There's a lot that I have to offer that'll improve your well-being. And by you supporting, it's a win-win situation. You get something out of it, and you can help support the channel. With that said, let's go to answering some of these questions that one of my subscribers on Patreon asked for some explanation on. So it's important to understand this concept of messenger RNA. All right, let's just start with the basics. In a typical setting in your cell gets signals to do gene expression. So, and there are cofactors through that signal that leads to some sort of DNA to unravel to start coding for RNA. And that RNA gets processed within the nucleus of the cell and eventually becomes messenger RNA that's ported out of the nucleus and into the cytoplasm to produce protein. And those proteins do certain functions for the cell. For it to port out of the nucleus, you need to have something called a 5' cap. All right. So that's this right here. All right. And that's kind of like a chemical structure that's on the RNA. And then you need a poly A tail at the 3'. And then your typical messenger RNA that your cell produces will have some structure to it. All right. Through these hairpins. These hairpins are created when there are complementary sequences that can kind of attach to each other. So an RNA is ACGU, while DNA is ACGT. T, right? G. So those are the nucleotides. So when you have complementary ones, like an A with a U or a C with a G, they can kind of fold onto each other and attach. It's called annealing. So that annealing process could take place, and then you can have these hairpins where there's no complement. And this three-dimensional structure is important for the stability of the messenger RNA, but also to hide regions so there is no coding. And then you have a start codon and an ending codon for your ribosome to come in and produce protein. It's also important to know that when the cell produces these messenger RNAs, it can make a lot of these messenger RNAs, and then it has lots of ribosomes floating around in the cytoplasm. So you can be producing lots of proteins in a short amount of time. In addition, you can actually have multiple ribosomes attached to a messenger RNA if it's long enough. And so you can have a start codon here, and the ribosome from the cytoplasm in the cell attaches and starts producing protein, right? And it's like a string of amino acids. So it's producing protein. And it gets near to the end, and another ribosome may be attaching. So each messenger RNA, depending on the length, if it's short, no. But if it's long, you can actually have multiple ribosomes attached and producing proteins, let's say three at the same time. So you're producing three proteins at the same time with just one messenger RNA. But if there's a strong signal for expression in the cell, it'll produce a lot of messenger RNA. And so you're going to be producing lots of protein in that cell. All right. That's the typical conventional non-amplifying messenger RNA. Now, let's talk about self-amplifying messenger RNA. Now, this could be, in technical terms, this could be in pathogens like viruses. All right. This was developed by this concept of having self-amplifying messenger RNA as a lab technique and a potential therapy technique for gene therapy was developed first in Japan. But there are lots of different labs that are playing around with this now. Now, what they did, they took the concept that what is happening with viruses, where some viruses will have, or many viruses have, a non-structural protein or proteins that will attach to themselves and create a complex that will help with coding or replicating the RNA. All right. So either coding protein or helping with replication. All right. But with viruses, it needs some of the machinery of the cell to do it. So in this case, usually what happens if there's non-structural proteins in the actual virus, it needs the ribosome in most cases, not all cases. There are some that have their own replicase built into the viron packaging. But the point here is that usually it takes a ribosome in the cytoplasm to start this off from a virus. Okay. So that ribosome in the cytoplasm of the cell would attach here and then produce this complex, these non-structural protein 1, 2, 3, 4, 5, or whatever, in this case 4. And a complex would be built up. And that complex is a RNA-dependent RNA polymerase that would attach to a promoter that will lead to producing code for a protein or an antigen. All right. So these researchers in Japan basically was just taking the mechanism that viruses do and slicing it in such a way where it potentially could be used as a therapy. So that's some background on that. So what are some of the similarities between the conventional non-amplifying messenger RNA compared to the self-amplifying or self-replicating? Some people call it self-replicating, self-amplifying. So the self-amplifying messenger RNA is what is the shorthand of that is SA messenger RNA. Okay, so you have what's similar is you still have the 5' cap. You still have the UTR, the untranslated region, the 5'. You still have the 3' UTR, and you have the poly A tail at the 3'. All right. You have code that you want a protein to be made from. Translated is called translation. The differences are that you have this non-structural protein code to create a replicase, an polymerase, an RNA dependent RNA polymerase. And the other feature is that you have some sort of promoter region that will have this complex attached to and then build up that protein. All right. So this gets into the cell somehow. We'll go through the details of that in the next slide. But this gets into the cell somehow. And it needs the ribosome from the cell to attach here and start coding for the proteins. And your cell's producing all these NSPs, and they self-assemble into a replicase or polymerase. So it'll and then that complex of proteins that your ribosome created or translated into your cytoplasm, they're floating around that complex, and it'll attach to this promoter region. And that promoter region will then that complex that these make up, these proteins that are made up, will attach and create a new protein that's the actual protein that you want for treatment. For example, let's say that you have a disease like cystic fibrosis where you have misfolding of proteins. But if you produce proteins in the cytoplasm that are properly folded, then that person's cell will not exhibit the disease of cystic fibrosis. So that's the reason why they were trying to use these types of techniques to try to produce protein. It's like gene therapy, but produce protein in the cytoplasm to help with a certain disease. Usually there's some sort of misfolding of the protein or a knockout. You may not even have a protein. And if you produce proteins in these cells, then you won't exhibit the disease or you mitigate the disease. So that's the reason why these researchers were looking at this. So they were taking things that are similar to viruses, slicing and dicing it and modifying in such a way where it potentially could be therapies for individuals that have protein folding problems or protein knockout problems, certain genes that are knocked out. Okay. Now, what we want to talk about here is I think most people realize this, but just to make it very clear to the ones that are listening, these NSPs, these non-structural proteins, 1, 2, 3, 4, you can have this is the code to create that protein section. So this is the RNA, this is the messenger RNA code that, if translated, will create a protein that would be an NSP1 protein, and then an NSP2 protein, and then an NSP3 protein, NSP4 protein, and those proteins would attach to them together, self-assemble, and then attach to the promoter region that would lead to producing the code that you need to fight a particular disease, let's say cystic fibrosis in this case. All right. So that's basically what it is. So the more you have of this, the better if you're trying to have high expression of a certain protein to fight a disease. Now, if you only have one of this in your cell, And we'll talk about how it gets to the cell later in the next slide. But let's just say you have just one messenger RNA. And the replicase that's per, or let me use the proper term here. The polymerase from this complex is produced and attaches to the promoter. Your antigen code that is, or the proteins that are being produced will be dependent on the concentration of the polymerase that was produced from your ribosomes, because you only have one messenger RNA that has this. In this one case, there are cases where I'll show you that you'll have lots of this. But if in your cell there's only one messenger RNA and it can't produce more messenger RNA, but it can produce lots of this in the NSP complex, this polymerase, then it's going to produce protein. And that the function of how much protein that would be produced is that the template is only one. You may have lots of concentration of the replicase because your ribosomes created lots of this polymerase, I meant. So the bottleneck is how fast a polymerase can attach to the promoter as it's producing code. So that first polymerase would attach to the promoter, starts producing protein for, let's say, cystic fibrosis protein. And then as it's moving along, another complex attaches, polymerase, and it's producing more protein. So now in this case, we have two strains of protein being produced, strands of protein. And then let's say this code is long enough where you can have another complex attached to the promoter, and it's now producing three off of just one template. That's the point I'm making, is that it's a function of how many templates you have and what's the concentration of your polymerase. If you only have one template and one polymerase, then it's a slow production of the protein. But if you have lots of polymerase and you have only one template, you can produce multiple copies depending on the length of the messenger RNA, multiple copies of the protein off of just one template at the same time. All right. And as one replicase falls off, another one comes on, and then it moves along to the mid, and another one attaches to the promoter. So you got two being produced at the same time. And then as it's moving along, if it's long enough, if this code is long enough, then another polymerase will come, and then a third or fourth or fifth protein strand will be produced. And the technical term is a peptide. So you can have multiple peptides being produced at the same time, depending on the length of the code. But there's going to be a point where even if you have very high concentrations of the polymerase, because of the length, you can't, it gets congested and it can't move because there's a certain speed that the polymerase is moving along the code and producing the actual peptide. But the point here is that if the code is long enough, you can produce multiple peptides at the same time. It's just that they're going to be at different lengths. The ones that are closer to the end of the antigen code would be longer peptides, and the ones that the polymerases that attach to the antigen code at the beginning would be shorter peptides. Okay, so let's just keep that in mind. So now the next thing, oh, uh, Trying to get the zoom off. I don't want this. I don't know why that is. Oh, there we go. Okay, so now we go to the next slide. Now, the next slide is taking it from the perspective of, okay, we have a self-amplifying messenger RNA that's in the cell. Now let's talk about how does it get to the cell? Well, there's lots of ways that you can get messenger RNA to the cell. You can have a lipid nanoparticle. You can have a VLP, a viral-like particle. You can have um, there are systems that are being developed where you could have an antigen signal or attachment on the cell, like a receptor, and you can have an antibody that attaches to that receptor, and that antibody is on top of some sort of lipid membrane, and then so you can have different versions of kind of like a viral like particle. Some of them have antibody type attachments to the particle. Some of them just have some sort of protein attachment to the particle that will dock to the cell. All right. So you could have VLPs, you can have LNPs. You can put them in actual viruses that have been designed as vectors to infect cells. So these are delivery systems. Another way to get to get these messenger RNAs into the cell is to actually transfect the cell with a plasmid, and that plasmid would have complementary DNA. And that DNA, once that plasmid gets into the cell, then what happens is that there are enzymes that are in the cell that will code the DNA complement plasmid into messenger RNA. And that messenger RNA would be then in the cytoplasm. All right. So there's lots of ways to get the messenger RNA in the cell. Lipid nanoparticles, viral-like particles, plasmids, or special designed vectors that are shells, certain types of shells of a virus, non-replicating virus. All right, so it gets into the cell, and eventually, somehow, some way, that messenger RNA is now floating around in the cytoplasm. All right. Now let's assume that that messenger RNA that came from the lipid nanoparticle or the viral-like particle or whatever mechanism, it has this self-amplifying code. All right. Now there are two scenarios here. One is a self-amplifying scenario, and then the other is just a regular-the-mill messenger RNA. Now, the regular mill messenger RNA could have been done by two methods. One method is that the nucleus gets the signal to create the protein. So the DNA turns into RNA that goes through a process that creates the messenger RNA. It ports out of the nucleus. It gets into the cytoplasm. The ribosome in the cytoplasm attaches to the start codon, and then it creates the protein or the antigen in this case. Right? It's just antigen is the same thing as the protein or the peptide. All right. So that's just the normal process. Another way that it could have came in was through a lipid nanoparticle or a viral-like particle or some sort of viral vector that was designed to get into cells and put messenger RNA in there. Another way is to have that plasmid, like I said, a plasmid that has complementary DNA gets into the cell and it eventually starts coding for messenger RNA. All right. And then that messenger RNA would produce the antigen. But again, that messenger RNA code, no matter what the vector was to get into the cell, to transfect the cell, is just creating the antigen with no self-amplifying capability. It doesn't have the NSPs, the non-structural proteins. Okay. So that's one scenario. The other scenario is that it has a self-amplifying messenger RNA capability. Again, coming from either some sort of plasmid, coming from a lipid nanoparticle, coming from a VLP, viral-like particle, or some sort of a viral vector that was designed to attach to the cell, but not self-replicate, but it has the messenger RNA code in it that allows for for um for the NSPs. It has in its genome. Okay, so it gets into the cell and it's now floating around in the cytoplasm. This messenger RNA. And that messenger RNA has the 5 prime, right? It also has the cap and all that stuff. All right. And then it has this NSP structure that will be what is called the RNA dependent RNA polymerase, the RDRP. Okay. And then they have this antigen code. Let's say in our case, the cystic fibrosis protein that's properly folded. Okay. So that's the code for the cystic fibrosis, let's say in this case. So what happens is that the ribosome in the cytoplasm will attach to the codon, the start codon, and start coding proteins for the NSP1, NSP2, NSP3, NSP4, and then fall off because there'll be a stop codon. All right. And what happens, really what's happening here is that there is a stop codon, there's a start codon and a stop codon, a little bit of space, then another start codon and a stop codon, little space, start codon, stop codon, then a little space, start codon, stop codon. So you have this ribosome from the cell, from the cytoplasm, that will attach to the start codon, code for NSP1, and then fall off. And then what will happen is it's producing many of these NSP1s, but you also have ribosomes that are attaching to the start codon for the NSP2 and creating multiple proteins of NSP2, and the same thing for NSP3 and NSP4. So you have lots of this RDRP that's produced. And then this RDRP, right, will, if it has a promoter region, could attach and produce the code for the cystic fibrosis if it had a promoter region for this RDRP, like we had in this case, this subgenomic promoter. Okay, but we want to take it and make it a little bit more, have a little bit more function. So when you have a positively sensed RNA, you can code for protein right away. But if you want multiple copies of this, it has to become negatively sensed and then be copied to become positively sensed again. So this RDRP has 2 functions. Sometimes it can be designed where the RDRP is designed to find the promoter region and create the code. Sometimes the RDRP is designed to actually reverse the code, make it from positively sensed to negatively sensed, and help with promoting. that promoter region. Okay, so depending on the shape of the complex, depends on what it can do. Sometimes the RDRP will attach to the promoter if it's designed that way. If it's designed to actually turn into negatively sensed, which is what many viruses do, then what you have is that you're going to take that positively sensed RNA, one copy template that came from, let's say, the lipid nanoparticle, and RDRP, many RDRPs are created from your ribosome in your cell. And what's happening is it's taking that template and it's turning it into a negatively sensed RNA. So it's all negatively sensed RNA that's coding for the opposite. All right. And then once that happens, then there's another RDRP that will come in and that's floating around in the cytoplasm. It's already been created from the first template, and it's producing lots of positively sensed RNA. To make the positively sensed RNA, you need a template that's negatively sensed. And that's the reason why the RDRP was taking one copy template from a lipid nanoparticle that transfected into a cell. And that transfection, one template creates the RDRP. Lots of different a higher concentration RDRP is floating around in the cytoplasm near this code. And that RDRP is designed to multiply the messenger RNA. So what's happening is that it's taken a positively sensed RNA, one template, only one messenger RNA, positively sensed, and making multiple negatively sensed RNAs. And those multiple negatively sensed RNAs can further be converted back to a lot of positively sensed RNAs. And that's how it's replicating the messenger RNA. You can't do that with this, because in the cell, when it's making cellular messenger RNA, it's using DNA and producing lots of messenger RNA that's ported out into the cytoplasm based on signals that the nucleus is responding to of how much antigen, how much peptide, how many proteins it needs to produce in the cytoplasm. So it doesn't need a self-replicating messenger RNA. The replication is the production from the DNA to RNA. That's in the typical scenario in the cell. All right. So, okay. So you have lots of this now messenger RNA self-replicating. And because you have this RDRP, lots of this RDRP produced, because when this is produced, lots of the positively RNA, more RDRP is going to be produced. And because the RDRP is going to be produced, it's going to be able to attach to the promoter region and make the code that it needs to make high concentration levels. And this is the reason why the concentration of the antigen is so much higher with self-replicating than non-self-replicating. All right, because it's making lots of copies of this messenger RNA. All right. Now, when you do that, let's say in the case of cystic fibrosis, you have a misfolded protein, all right, and that causes the cell to not function properly and show the signs of, let's say, cystic fibrosis. But if you had a self-replicating messenger RNA template that gets transfected into a cell, and it had this structure where it had this RDRP, it would take that template, convert it to negatively sensed RNA, produce lots of copies that used to be just one template into hundreds, maybe thousands, depending on, what you're doing here. But let's say in our example, hundreds of this. And it has lots of RDRP that's produced. There was a smaller concentration from the first template, but now because you have 100 of the positively sensed RNA now, you're going to have a higher concentration of the RDRP. So it's going to attach to the promoter region. In this case, the promoter region, the subgenomic promoter region. And it's going to produce lots of RNA. I'm sorry, lots of protein. All right. Lots of antigen, lots of protein. Because it just has more template compared to the concentration that was governed by how much the DNA converted or translated into RNA and ported out as messenger RNA. So you can see here that self-amplifying will produce lots more than the typical example. Now, remember, this example also could be through through the lipid nanoparticle issue that we dealt with in 2020. All right. I have to be careful the way I say this. But you'll understand if you read in between the lines of how I'm phrasing this. All right. So this template for messenger RNA that was coming from the lipid nanoparticle in 2020, it did not have self-replicating. It did not have the NSPs. It only had the S1 and S2 subunit, and then produced the S1, S2 subunit that would be transported to the MHC2 to activate the CD4. All right, to start the antibody process to produce antibodies. All right. Now, the problem comes in is that what happens if it's reverse transcribed and it gets into the cell and it produces lots of it. All right. Then it goes into a governed DNA to RNA concentration with growth signals. As long as it hits the promoter region, it can produce lots of this. But this doesn't have the self-replicating part to the code. It only had the S1 and S2 subunit for what I'm talking about for 2020. So that means that it did have concentrations, high concentrations of the antigen, but a cell that actually been hit by a lipid nanoparticle at the very beginning, it only had one template. Now that template could be increased in concentration if there was some sort of reverse transcription that took place in the cell, and then the cell continued on making this because it kept on getting a signal to make it. Okay. But with this concept, you wouldn't need the integration. With this concept, this could produce lots of this protein without cellular integration, genomic integration through reverse transcription and integration. All right. Integrase is what's the actual enzyme. So there's reverse transcription, there's the integrase, and then boom, it gets into the cell. Now, another problem that happened with the 2020 was that you had plasmids that were able to get into the cell and then get integrated into the nucleus and produce lots of these proteins. All right. But also, if you had a plasmid floating around in the cytoplasm and you had a polymerase that could take that cDNA and convert it to messenger RNA, you would be producing lots of messenger RNA in the cytoplasm independent of the nucleus. So there's many different ways, many different problems that seem to be happening with the 2020 incident. All right. Reverse transcription, and integration, plasmid getting into the cell and producing lots of the messenger RNA, and that plasmid integrating into the nucleus, allowing for, if it was by a promoter region and it's activated, then it'll produce lots of messenger RNA. So that's the problem that was going on with the 2020 incident. All right. So, okay. So you got some problems if you have reverse transcription, and you have some problems if what you give isn't filtered out and there's plasmids in it. Now, again, the plasmids can get integrated with the nucleus. The plasmid could also produce RNA in the cytoplasm. So you can have an RNA, what is called an RNA dependent. I'm sorry, it would be called a DNA dependent RNA polymerase. All right, so with that said, now let's take a look at the more complex version of it, where you have all these NSPs. Well, you have this code, right? And this could get reverse transcribed, just like this could get reverse transcribed and get integrated into the cell and cause a problem. All right. Um, but let's say if it's cystic fibrosis, for example, if this gets reverse transcribed, this code, and it gets integrated in, and it can be promoted, then the nucleus is going to get those cofactors and that promotion attachment, and that would lead to more messenger RNA that would create the cystic fibrosis protein therapy. So in that case, that's actually good. All right. Now, because those cells don't have the proper folding for cystic fibrosis gene, this does have the proper folding. So if it gets integrated into the nucleus, that would be beneficial to someone that has cystic fibrosis. But if this is something like for a vaccine and it gets integrated in, that could be potentially disastrous. Or if this was some sort of bioweapon, that could be disastrous. So here's another example where this type of technology could be used for good or for something that could harm. All right. So keep that in mind. So you have this messenger RNA that could get with these NSPs that could get integrated in to the DNA. That DNA then gets a signal to produce more messenger RNA that's of this. And then what happens is it self-replicates in the cytoplasm and produces lots of protein. And if it's cystic fibrosis, guess what? It's most likely a good thing. All right. Because it would help if it was cystic fibrosis proper folding gene, then you're helping with someone that has cystic fibrosis disease. But if this is a vaccine, then you're producing lots of antigen, and you most likely are going to start having problems with your immune system. All right. Either potentially autoimmune disorder, or you're activating you're activating the immune system so many times that those signals may actually be immunosuppressive. Okay. Now, another thing that I want to talk about here is the member on Patreon was asking, should we be worried about this? What are some of the things to worry about, or what are some of the, could it be shedding or some these kinds of questions? All right. Can this technology be good for people that have problems with disease? This technology would be beneficial potentially to treat these types of diseases like cystic fibrosis, even sickle cell, or even Alzheimer's for that matter. All right, because there's a misfolding of protein, and if you can fix the misfolding of the protein and produce more regular protein, then you're slowing down or mitigating the disease. But if you're using it as a vaccine vector, then you're increasing the probability of vaccine injury, and you're increasing the probability of of and there's different types of injury, autoimmune or weakening of the immune system. All right. Not just that, depending on where it gets integrated in the nucleus, because you can't control that, it could disturb tumor suppressor genes. And the ability for that cell to fight cancer, a certain cancerous pathway, is diminished. So cancer incidences would probably increase if there was nucleus integration, if there was this genomic integration from this self-amplifying messenger RNA. Now you can design weapon systems where you're creating, there are oncogenes that we have in our cell that are usually turned off. But what if you create a system that allows them to turn on and turn cells into cancer? And you have a messenger RNA with the NSPs that are, self-amplifying, that help to turn on oncogenes or turn off tumor suppressor genes. So you can design this in a way where you're creating lots of code to activate or inactivate certain things in the cell, which would mean that you can use this as a weapon system. So again, you can use this for nefarious reasons or to try to treat cystic fibrosis. And this is the real problem with people playing around with this stuff. This is that if you got the wrong actors, if you got a military industrial complex playing around with this stuff, you can do some really nefarious things with the nucleus and with the cell and the cell processes. All right. Or even the endocrine system or the immune system. So just keep that in mind. So there's a danger there, but there is a potential benefit if someone has Alzheimer's or cystic fibrosis or some other protein folding problem, or some sort of lack of protein or low concentration of a protein. So it can be harmful, and it could be also beneficial depending on how it's used. Now, to answer the question, should we worry about it amplifying to such a degree where it's going to spread all over the body? No, I don't. This is going to be at the cellular level and at the tissue level. And then there are feedback loops within your cytoplasm where it knows that if it has too high of a concentration of a messenger RNA, it will actually use RNase to break it apart. And if there's too much protein, it also activates proteases to break up protein. Now, the thing is that this gets back to the nefarious part of it. If you can produce a messenger RNA in non-structural proteins that not only are DrPs, but also proteins that inhibit. So you can, let's say, for example, you can create NSP1, NSP2, NSP3, NSP4 DrPs. And you can also have NSP, let's say 5, 6, 7, and 8. And those NSPs self-assemble and turn off the protease. If it turns off the protease, then you can't break apart high concentrations of antigen that are produced. Also, you can have another series of NSPs where you can have NSP 9, 10, 11, 12 that turn off the RNase, that inhibit the RNase to prevent the RNase from breaking apart the RNA. So if you design systems that act like viruses, you can shut off the backup systems to try to prevent high concentrations of this stuff in the cell. The nucleus, the cell, has signals that will produce proteases and RNases. But if you inhibit the proteases and the RNases, then you've hijacked the cell and you can produce a lot of self-amplifying messenger RNA, and you can produce a lot of antigen that it's coding for. So it's a system that can turn off certain pathways of the cell that try to that so it's like hijacking the cell, just like a virus does. So go back to, we have different mechanisms in our cell to try to prevent cancer. Tumor suppressor pathways. There are lots of things have to be knocked out before a cell actually goes cancerous. Depending on the cell, it's like 5 different knockouts have to take place. Some cells do have a 3 knockout. Humans need about a 5 knockout process. Mice or rats, rodents, there's usually a 3 knockout process that'd be cancerous. So the point here is that if you produce NSPs that shut down the tumor suppressors, if you have NSPs that turn on oncogenes, if you have NSPs that are turning off proteases, if you have NSPs that are turning off RNases, then what will happen is that you can infect a cell and turn it cancerous. That's the problem. Now, what about shedding? Let's talk about the shedding problem. If the messenger RNA code doesn't have structural proteins to create a VLP, then shedding would be low probability. Because even if you had shedding, let's talk about what is shedding. Well, there is this concept that the laymen have where it's shedding from one body to another body, human to human. Well, there's also shedding at the cellular level, right? So let's talk about the shedding of it at the cellular level. Shedding at the cellular level would mean that you have, let's say, an RNA. I'm going to run different scenarios here. You have an RNA with self-amplifying that gets leaked out of the cell and is in the cellular matrix or in your bloodstream. Eventually, because messenger RNAs, if they're not wrapped in some sort of lipid system, a nanoparticle or a viral-like part, an exosome, whatever, then it will degrade. It's not stable if it's not in the cytoplasm. So if it's in just the cellular matrix, it will eventually degrade. So there is shedding out of cells, but it will degrade. Now, let's say the cell is necrosed. There is necrosis of the cell, and there's lots of leaking into the extracellular matrix, and you have lots of this stuff. So you will have a lot of shedding, a lot of leaking into the cellular matrix. Now, some cells adjacent to it might bring in some of these RNAs that are not wrapped in, let's say, an exosome. Well, so there is some shedding that's coming out of the cell and getting into other cells. And when it gets into other cells, it can self-replicate. But that mechanism of RNA in the cellular matrix getting integrated into an adjacent cell does happen could have some sort of runaway effect, but eventually it kind of dies out. There may be a runaway effect at the beginning, and then then there's your system is activated, assuming that your immune system or your innate immune system is activated in a way where it kind of walls this off and then starts killing off the cells, and this process starts to go down and the self-replicating stops. All right. But the problem comes in is that you do have this can get wrapped into an exosome of the cell. And that cell's exosome could transport to an adjacent cell and then infect, and it would be self-replicating. So for cystic fibrosis, instead of having multiple injections throughout the year to try to have this get into the cell, if you design it in a way where it can be wrapped in the exosome process, then you do one or two therapies, and then the cells adjacent will get transfected from the exosome that will lead to treating cystic fibrosis. But let's take the point of a bioweapon. If you have this messenger RNA that's in the cell that can get wrapped up in an exosome or a VLP, then that VLP is going to go to adjacent cells and transfect, and then boom, you have a spreading of a weapon system, a spreading of, let's say, a turbo cancer or whatever. Okay, so self-replicating can be very dangerous, but it can also help people that have severe diseases. It just depends on how you use it. Now, that's the leaking or the shedding process, if you want to call it that, at the cellular level. Now, at the human-to-human level, is it possible for an RNA that's in the cellular matrix getting into the bloodstream and somehow, fluid transmission or, blood-to-blood transmission, could it you possibly get the RNA into another person? Yeah, it's possible, but it's highly unlikely because the messenger RNA is unstable in that environment. So it's not even going to work. It's going to break apart. Like, for example, it's not going to be that stable if it's not in an exosome or in a lipid nanoparticle or some other packaging. Because what will happen is this RNA, while it's floating around in your bloodstream or in your cellular matrix, it's going to break apart. It may fracture, let's say at the NSP1 or, between NSP2 and NSP3 and break apart. And if it's broken apart and you happen to get it, it doesn't mean anything. It won't do anything. Because that RDRP needs NSP1, NSP2, NSP3, and NSP4, and the promoter region, and the code, and the antigen code. So this thing is just going to start breaking apart in the cellular matrix or in your bloodstream. So it's not going to really. Will there be some shedding? Yeah, you could get pieces of this, but it won't mean anything. But if it's in a lipid nanoparticle, or if it's packaged in some sort of viral-like particle from the cell or an exosome, then the transmission to human could happen with a stable messenger RNA packaged inside. That's the whole reason why the exosome and endosomes are so important for stabilizing proteins and stabilizing transporting of RNA. I think what will happen in the near future is more research is going to shed light on not only are proteins being shuttled back and forth between cells in tissue, but also RNA. So, and that's just a natural process. That's just how the body works. But I think more research is going to shed a light on that through this endosome exosome process. So could you get a shed from a self-amplifying messenger RNA that someone was treated or someone was infected with, and it's packaged in an exosome? The probability is more that it will just shed and for that individual that has this and just spread in the body or in that tissue. But it could potentially be shed from human to human because it's more stable with a lipid layer around it. If it's not packaged in a lipid layer, then the messenger RNA is not going to be stable. And even if you did get pieces of it, of the shed, theoretically, it wouldn't do anything. So it wouldn't matter. But the problem comes in is that if it was wrapped in a lipid, if it's wrapped in a lipid, then you're talking about something that is a little bit more like close contact, touch, sharing of fluids, that kind of thing. Now, if you really want to get crazy, you could design the stuff to be respiratory. But that's, that's a little bit more involved in doing that. But it's possible to do. But the point here is that this self-amplifying messenger RNA is taking a piece of a mechanism that is actually viral, a viral mechanism, and using it as a weapon or using it as a therapy. So to answer the question, should we worry about this? Well, it could be used as something for treatment, and it could be used for nefarious reasons to cause harm. Cystic fibrosis is a perfect example of how it could help. Another thing is how it could help with people that have cancers is that you could actually create antigens that actually shut down these fusion proteins that are causing the cancer, and then have the cell to actually go into apoptosis and die off. So that's the benefit for people that have cancer. But you can design this in such a way where you're shutting off the tumor suppressor genes and you can promote cancer. That's being used for nefarious reasons. The shedding concept, I'm not so worried about because the transmission would have to be very close contact and it would have to be wrapped in a lipid nanoparticle. Some sort of lipid layer wrapped or that's wrapping around this self-amplifying messenger RNA. Because if the shed was just the messenger RNA without the lipid membrane, it would break up. It would break up pretty quickly. It's not stable. So the question is, how often does that happen where it's integrated in an exosome in bodily fluids, and then those bodily fluids are transmitted from one human to another. So now we're looking at what kind of transmissions for shedding. We're looking at something that's like HIV. We're looking at something that's like Ebola or Marburg or pox viruses. But the average person, on the train or, in the grocery store, I would not worry too much about self-amplifying messenger RNA if they're not in a lipid protective coating. If they are in a lipid protective coating, then you could get a self-amplifying messenger RNA. But the probability of it is low, but it can happen. So hopefully that gives you a better understanding of what is going on with self-amplifying messenger RNAs. It's basically taking a piece of a viral mechanism and playing around with it to get more antigen in the cell, to produce more protein in the cell. And depending on what you want that protein to do, depends on if it's going to be for positive or for negative reasons. Will it be harmful or helpful? So just to go back, one step here in my thinking here. So you have this one template that transfects the cell that's positively sensed. It's producing the RDRP. That RDRP is producing lots of negatively sensed code that can't make protein. But also this RDRP is making some antigen, but not in high concentration because there's only one template. But now it's created a negatively sensed, and that RDRP can also reverse that and make lots of positively sensed now. So now instead of having one template positively sensed and just a little bit of antigen, it has lots of positively sensed code and lots of RDRP, lots of promoter regions that can produce lots of antigen. Now, you can also design this in such a way where the RDRP in the negatively sensed um, template, you're going to produce a lot of negatively sensed template. You had one template originally of the positively sensed. The RDRP is produced. It's producing lots of negatively sensed or of all right, and you have these RDRPs, right? That that technically could bind to a region in the negatively sensed template and produce the messenger RNA code that's positively sensed and only the antigen code. All right, so let me repeat that so people can follow me here. This thing can go two different ways. Once you have the negatively sensed RNA from this reverse, from positively sensed to negatively sensed RNA, you have lots of this template. But this RDRP can be designed in such a way, or the reverse, this RDRP could attach to a promoter region and produce and read from 5' to 3', right? And produce. Let me rephrase that. There's two ways that this thing can happen. You have the positively sensed RNA, one template. Lots of RNA negatively sensed can be produced from this RDRP, this complex of the NSP1 through NSP4. So you have lots of this. And one way is that, okay, that RDRP then takes lots of that negatively sensed RNA and produces lots of positively sensed RNA. And because there's a promoter region, it can also produce lots of antigen code. All right. But you can design this in a way where this RDRP recognizes a promoter region that's negatively sensed. All right. That would have been and code or an open reading frame. Let's call it an open reading frame. And it'll sense this here. And this 5' to 3' code reads from this direction. Okay. So to produce this, that you have one template, 5' to 3', right? To reverse it, it's going to be 3' to 5'. So you can have a region that's actually coding more messenger RNA that's just antigen code instead of having the NSP part of switching back from negatively sensed to positively sensed is the point I'm making here. You can have a region here that can actually start coding messenger RNA versus coding for all of it. So you're going to have some that are going to code for all of it. And you're going to also have a region that the RDRP can recognize to actually produce just the antigen code. That's the point I'm making here. All right. Now you can get even more sophisticated and and it can do other cellular processes on top of this. But we're going to try to just simplify it here for a second here. But it's really important to understand that self-amplifying kind of overrides that check system that the nucleus normally has with certain signals, feedback signals, to produce messenger RNA. And then if it gets that signal to produce more messenger RNA, it'll produce it and then antigens created. But it also has a signal to say stop making it. And also it gets signals to produce proteases to break up the proteins if there's too much of a concentration. And the nucleus also, and the cytoplasm, has signals to produce or to activate RNase to break up too much of RNA that's floating around in the cytoplasm. But if you design a NSP series within the self-replicating, so you can have a self-replicating and an inhibitory non-structural protein complex that turns off the RNase and or the proteases. And then the cell can't stop this. It shuts down, it hijacks the cell. So should someone worry about this technology? Yeah, someone should worry about this technology. Is it highly probable that it's going to, that it'll shed and it's going to spread all over the world and turn, gray goo? No, that's not going to happen. But that's very low probability, let's put it that way. But can shedding happen? Yeah, shedding could happen. If it's just the RNA shed, it's not going to be stable and it won't matter. But if it's wrapped in some sort of lipid from the cell, it's going to be more stable and could spread possibly from human to human, but definitely from cell to cell within the original patient or the original person that had this self-replicating messenger RNA. So hopefully I've explained it so you can see some of the mechanisms that this is happening with. It's borrowing from mechanisms that are viral. But the more you turn this into other viral mechanisms, not just the self-replicating, but you can turn off certain proteases or turn off certain RNases, then you're hijacking the cell and now you're turning a self-amplifying messenger RNA into a human-made virus. That's the problem. Okay, so please help support my work by going to my website, the-studio-reykjavik.com. Take a look at the different products that I offer. And I do these videos for free. It takes time to make these videos. Please help support by purchasing products and donating on my website, the-studio-reykjavik.com. You can donate through Stripe, PayPal, or buy me a coffee. You can be a paid member to my Patreon channel. And you learn a little bit about what's going on in the sciences. So please help support my work. I have thousands of videos for free. And share the links and ask your social media to follow me. Thank you for listening and have a nice day.